Good morning, everyone. It's an honor to have the opportunity to present my work in this conference and to address such a distinguished audience. I'm Maalej, a PhD at Institut Supérieur de Gestion de Tunis from Tunisia and attached to Best Mode Laboratory. In this talk, I would like to present our study entitled Risk Factors of Breast Cancer Determination, a comparative study on different feature selection techniques elaborated by myself, Dr. Kaouther and Dr. Fahmi. We have organized this talk in the following headings: context, problematic, objectives, contribution, experimental results, and finally, conclusion and future works. Artificial intelligence includes several techniques that allow digital machines to simulate the behavior of human intelligence. In this context, machine learning, which is a subset of artificial intelligence, has shown considerable performance in several areas such as speech recognition, natural language processing, text understanding, cybersecurity, computer vision, and more specifically, medical informatics. In this field, there are several analytic tasks that have been studied in the literature, such as prediction of the next hospitalization, prediction of all diagnostic categories for the next visit, and specifically disease prediction and monitoring. This can include cancer, heart illness, Alzheimer's disease, and Parkinson's disease. It is within this context that our subject fits, more precisely in breast cancer. According to the World Health Organization, in 2020, breast cancer affected approximately 2.26 million people worldwide, resulting in about 627,000 of deaths. It stands as the fifth leading cause of cancer-related mortality and the most common cancer among women. Our main problem lies in the difficulty of effectively identifying the true risk factors associated with breast cancer. This challenge can lead to misdiagnosis and delayed treatment, which directly impacts patient outcomes and survival rates. For this reason, there is an urgent need for more efficient methods to accurately identify risk factors and to enhance predictive performance in clinical practice. Despite the large number of studies addressing this problematic, the literature still shows considerable challenges. First, many studies rely on limited or heterogeneous datasets, which causes variability in the features that are selected. Second, the feature selection methods themselves are often unstable when the data changes. Finally, this combination of limited data and unstable methods reduces the overall reliability of predictive models, making it difficult to draw consistent conclusions or improve clinical decision-making. This creates a gap that limits the reliability of current predictive models and reinforces the need for more robust and systematic approaches. To overcome these difficulties, we aim at identifying the risk factors and enhance predictive performance for breast cancer disease, which will benefit all the stakeholders. For patients, it will improve their health status and can delay the progression of the disease. For physicians, it helps in recommending the appropriate drugs, and for hospitals, it helps in reducing medical costs. Unlike previous studies with limited data or unstable feature selection, our work systematically evaluates multiple feature selection methods on real datasets to identify key risk factors. We assess their stability under dataset changes and test classification performance. For this study, we used the breast cancer Quembria dataset from UCI. It contains 116 patient instances with clinical and demographic attributes, including age, BMI, glucose, insulin, HOMA, leptin, adiponectin, resistant, and MCP1. The target variable is binary, with 1 representing healthy patients and 2 representing cancer patients. This dataset provides a well-structured foundation for analyzing risk factors and evaluating predictive models. Our proposed method consists of different steps. First, we use the raw clinical and demographic data as input. Second, we applied various feature selection methods, including filter, wrapper, and embedded techniques. After that, we performed comparison and stability analysis under dataset perturbations. Finally, we used the selected features to build predictive models with support vector machines, logistic regression, and decision trees, allowing us to evaluate their performance and reliability. We used 3 categories of feature selection methods. First, filter methods including chi-square, which measures relevance for discrete variables. F-test, which assesses feature importance via group variance. And Gini index, which quantifies attribute impurity. Second, wrapper methods such as recursive feature elimination, which iteratively removes the least important features. Finally, embedded methods perform feature selection directly during the model training process, integrating feature evaluation with the predictive algorithm itself. To evaluate the stability of selected features, we used 2 metrics. The Jacquard index measures the similarity between 2 sets by comparing their shared and total elements. The Spearman correlation assesses how well the rankings of features match between different dataset perturbations, giving insight into the consistency of the selected methods. For classification models applied in this study, we used support vector machines which find the optimal boundary separating classes by maximizing the margin between them. Also, we used logistic regression. This model predicts a binary outcome using a probability model based on the sigmoid function. Finally, decision trees were performed which aim at splitting the data into branches based on feature rules to make predictions. For our experiments, we selected Python as the programming language due to its extensive libraries for data analysis and machine learning. We conducted all computations within the Anaconda Jupyter environment, which is an open source platform that provides an integrated and efficient workspace for data science. Using filter-based methods, no features were removed from the BCC dataset, although the ranking of features varied across methods. The wrapper method identifying glucose, resistin, HOMA, and BMI as key features, while age, adiponectin, insulin, leptin, and MCP1 were less relevant. The decision tree eliminated only MCP1 and highlighted the importance of glucose and resistin. Overall, glucose and resistin emerged as the top-ranked features across all methods. Their biological relevance is well supported as hyperglycemia can increase breast cancer cell proliferation, and resistin promotes cell growth, invasion, and migration. In summary, glucose and resistin appear as robust and significant biomarkers for breast cancer risk in our study. To evaluate the stability of feature selection algorithms, we used first the Jaccard index. All methods achieved a Jaccard score of 1, indicating that they selected exactly the same features across datasets perturbation. This shows that the methods are not sensitive to noise in terms of feature presence or absence, highlighting their robustness, which is not the case when using Spearman correlation metric. Chi-square is the most stable with a perfect correlation of 1. F-test and Gini index are moderately stable, while decision tree and recursive feature elimination show low to negative correlations, meaning their rankings are highly sensitive to data changes. In summary, all methods select features consistently, but their rankings vary, with chi-square being the most reliable and recursive feature elimination and decision tree the least stable under data perturbations. We compared the performances of SVM, logistic regression, and decision tree using all features and then selected features from different feature selection methods. We observed that for SVM, the accuracy remains the same at 0.91 regardless of feature selection, showing that the selected subset preserves performance. Logistic regression drops slightly from 0.78 with all features to 0.75 with selected features. This indicates a minor sensitivity to feature reduction. The decision tree improves from 0.92 to 1 with selected features, highlighting that feature selection can significantly enhance performance. Overall, feature selection impacts each model differently. To conclude, we applied multiple feature selection methods to identify the key risk factors for breast cancer and evaluated the stability of these methods under dataset perturbations. We also compared classification performance using all features versus the top-ranked features. Our results show that the decision tree is particularly effective in both feature selection and classification. The most important risk factors we identified include invasive status, glucose, resistant, insulin, leptin, age, adiponectin, BMI, and HOMA. Therefore, our work provides a robust framework for early breast cancer prediction, offering a reliable tools to support clinical diagnosis. Despite the notable performance achieved, this study has some limits. First, it is based on a single data set, so the results may not generalize to other populations. Second, although we evaluated stability, feature selection methods can still be sensitive to small changes in the dataset. Finally, we only tested 3 classification models, and exploring additional models could provide further insights. For future works, we plan to validate our approach on larger and more diverse datasets to improve generalizability. We also aim to explore more robust feature selection techniques that are less sensitive to data perturbations and to test additional classification models, including ensemble methods, to enhance predictive performance. Thank you for your attention.